Hello, so welcome to chapter 21, dealing with the lymphatic system. For this chapter, instead of pulling the highlights from the normal PowerPoint presentation that is based on the flow of the book, I redid the PowerPoint so that it puts everything together with the different cell types and their actions all together in one section. So each cell type is going to have its own section, so everything just flows from how those cells are born, how they go through their maturation process, and what actions they go through. So starting with chapter 21, I went a little creative with this one, so yes, it's kind of corny, but it helps. So your body's immune response is based on the actions of immune cells, which we'll cover in this PowerPoint presentation, and then later your antimicrobial proteins that also act on behalf of your immune response. So here comes the corniness. Everything's based on a kind of high school imagery. So we're going to lymphatic system high, with our rivals being pathogen high, so any kind of invaders. So, sorry you can't see that. I apologize for any negative flashbacks, but it worked for grouping together all the different cell functions. Okay, so in your lymphatic system, there's two types of immunity. You have your innate immunity, where your immune cells just recognize anything that doesn't belong to the body. So if you remember the glycocalyx that we've covered earlier, if that glycocalyx does not match the exact same glycocalyx of the body, then those cells will recognize that difference and destroy that pathogen. So in general, it's kind of like saying you all can't sit with us because they don't match what is within the body. For adaptive immunity, it's where you either have receptors on the immune cells themselves, so your cellular immunity, or antibodies that flow through your body fluids, or also old school term humors of the body, to recognize an antigen, bind to it, then which gives it a destruction tag, and then destroys the pathogen. So again, your innate immunity destroys anything that's not a part of the body, so the glycocalyx does not match. Adaptive immunity is where you have receptors on these cells or proteins, which are your antibodies, that circulate through the body, and once they bind to a particular pathogen, causes the immune response. Okay, so for our actors in the immune response, they're broken up into three different groups. So you have first your upperclassmen, is what I call them. That's your lymphatic cells. Your freshmen are your leukocytes, so all of those 5 types of white blood cells. And then lastly, your antimicrobial proteins, or journalism is how I categorize them, also play a role. So we'll start with our upperclassmen, then move to our freshmen, and then go to our journalism group. So starting with the upperclassmen, or your lymphatic cells, there's 6 types of lymphatic cells, and they also have their own groups. So your NK cells, your T cells, and B cells are all type of lymphocytes. So they're like your power people in school. Then you also have your phagocytes. So these are the cells that will phagocytize or intake an invader and then destroy it. So bind with the lysozyme and then gets rid of the debris. And then lastly, by itself, you have the reticular cells, which help to maintain structure in your lymphatic tissues. So your first cell, a part of your powerful lymphocytes, is your natural killer cells. So I call them like a queen bee of our lymphatic body, since they are the least abundant lymphocytes. They're part of the innate immunity squad. So these cells, if they recognize anything that is not of this body, so of your body, so not matching that glycocalyx, then they will bind to that and attack and destroy. So that's their general purpose, is just to attack and destroy. So how these cells do that is they'll attach to the pathogen. They'll release a type of protein called preferins, which I call her first set of minions. They will create a hole in the pathogen. Then the NK cell will release another type of protein called granzymes, which are a type of enzyme that degrade protein. Those enzymes will flow into that hole, into the pathogen, and start degrading the protein, which causes this cell to undergo apoptosis or programmed cell death, because of the high level of damage from the granzymes. So that's how it gets rid of that pathogen. Your next lymphatic cell is your T cells. So again, this is still one of your lymphocytes. So for your T cells, I call them like student council. They're your most abundant lymphocyte, part of your adaptive immunity squad. So it has receptors that are very specific to a pathogen. So unlike your innate immunity, where if it just doesn't match, you're not welcome here, with your adaptive immunity, it's specifically targeted to something that has invaded the body. So it's also part of your cellular immunity. So here, instead of the specific binding coming from an antibody, it's coming from a receptor on the cell itself. So after a T cell has undergone co-stimulation, so when it binds, it must bind twice. So bind to the antigen once and then bind again. Once it's co-stimulated, then that triggers your T cell to make an army of clones, where it will make your 3 types of T cells. So there's your T cytotoxic cells, your T helper cells, and then your TM or your T memory cells. So with your T cells, your cytotoxic and helper cells, they actually undergo part of the attack phase of an immune response. And then your TM or T memory cells are part of your remembering the infection. With your T cells, they're not just born and ready to go. They're born in your bone marrow, and then they're educated in the thymus, so kind of like sent off to boarding school. Then after they have passed their test in the thymus, they're deployed to your different lymphatic tissues. So your spleen, tonsils, lymph nodes, anywhere where there's a high concentration of lymphatic tissues, where they'll can congregate and be able to fight an infection together. So after your T cells are born in your bone marrow, they're sent to the thymus to be educated, so they're tested for a proper response. So this is an adjustment from what I said in class, because it wasn't really clear in the book, but I kept going over it, and I cleared it up. So their first test is through positive selection. So first, the T cells must develop the surface antigen receptors, which allow them to become immunocompetent. So these surface antigen receptors are specific to different pathogens. So it's just by random genetic chance that they develop the right shape of receptor that will bind to a pathogen. After your T cells develop the surface antigen receptors, they're presented with self-antigens, so antigens that are matching the glycocalyx of the body where they are located. So these T cells cannot react to those self-antigens. If they reacted to the self-antigens, then those T cells would end up attacking the body once they're released. So if your T cells do not react to the self-antigen, then they're deployed into your naive lymphocyte pool, where they're later deployed and allow them to be exposed later to a pathogen, which then will activate them. If a T cell reacts, it has failed this test, and that T cell is then later deleted by negative selection. For negative selection, the failed T cells are eliminated either through clonal deletion or anergy. For clonal deletion, the failed T cells are phagocytized, so they're encapsulated or brought into a phagocyte and then they're destroyed. So this deletes that random chance clone of that particular T cell, so the shape of that antigen, and that clone, since it does not match correctly, so maybe it's too close to the self-antigen, that's why it's caused the deletion, that clone is deleted out of the pool of potential T cells to be deployed into the body. For anergy, the failed T cells are made inactive. So once a T cell passes the positive selection test, they're given a signal that continues them being able to live. If they fail the test, then one way of deleting them or getting rid of them is to deny them that signal that continues them being able to survive or live. And so those failed T cells are just made inactive. So they won't be able to respond to any of the antigens on your cell surface, since they reacted to them. Okay, so after your T cells are educated in the thymus, they are deployed into your different lymphatic tissues. And once there, that is when they'll be activated upon exposure. So with your T cells, you need an antigen presenting cell. So this is a cell that has encountered a pathogen, has phagocytized that pathogen, and has phagocytized it, then digested partially and presented part of that pathogen on its surface, so on one of its MHC proteins. That antigen obviously does not match the, or antigen of a pathogen won't match its host, so whatever's in the body, so the natural part of natural glycocalyx that is in the body. So when a T cell encounters that different antigen on one of those MHC or major histocompatibility complex proteins, once your T cells encounters one of those proteins on that antigen presenting cell surface, it'll bind to it. Then it has to bind again with a co-stimulation protein. Once that T cell is co-stimulated, then it's activated to become either one of three types of cells. So you'll create T helper cells, T cytotoxic cell or TC cells, and then TM or T memory cells. So you go from one cell to creating three different types of your T cells. So your first type of T cell is your T helper cell, so TH cell for short. So these cells will bind to the antigens on an antigen presenting cell. So here we have the antigen presenting cell. It displayed that antigen on its surface, so this T helper cell binds to it. Your helper cells, their job is to help out other immune cells in the body. So they secrete a substance called interleukins, which have 3 different jobs. They either attract neutrophils or natural killer cells through chemotaxis, so the increasing concentration of those interleukins help to attract those cells to the particular infection site. You also can attract macrophages to the area, so stimulating phagocytosis, where in taking into the cell and destroying whatever pathogen. Also, it prevents them from leaving the infection site, so keeps all those macrophages there in that area to do their job. Then lastly, it also stimulates T cell as well as B cell mitosis and maturation. So undergoing mitosis so you can make that clone army to make a bunch of cells that are specific toward that antigen, so against that particular pathogen, as well as going through the maturation process. For your next type of T cell, that is your TC cell or cytotoxic cell. Once these cells bind to antigens on a pathogen, so they're particularly the MHC1, excuse the typo, MHC1 protein, they'll bind to foreign antigens on an enemy cell. That T cell will then dock onto that cell, and just like your NK cells will release the preferrin proteins to make the whole, as well as release granzymes that will degrade the proteins in that pathogen cell. So these help to destroy that cell. It also secretes interferons to inhibit viral replication, which also helps to recruit and activate macrophages, so prevent your viruses from making multiple copies, and then those macrophages are brought there so that they can phagocytize them and get rid of those viruses. Then lastly, it will secrete the tumor necrosis factor. So again, macrophage activation to target the cells that are abnormal because they have become cancerous. So your last T cell is your TM or T memory cell. So these are TC, so T cytotoxic, or TH cells that remain in circulation after an infection. They're more numerous than your naive lymphocyte pool, since you've made that clone army after the T cell was activated and you undergo mitosis to make a bunch of copies of that cell. So you have a bunch of copies of that cell that's already specifically targeted toward a pathogen that you have been exposed to. So they're ready to have a quicker reaction upon re-exposure. So if you get an infection from the same type of pathogen, you have a bunch of these memory cells that are in circulation throughout your body, so they're able to have a faster reaction since they have less steps of having to go through co-stimulation and mitosis. You already have a bunch of them already ready to go to fight off that infection. Okay, your last upperclassman is your lymphatic cell. Upperclassman, still a lymphocyte, but this is your B cells. So your B cells, I make them kind of like your homecoming committee, where they can do stuff, but they need your student council or your T cells help. Still part of your adaptive immunity, so targeting a specific pathogen. But unlike your T cells, which are part of your cellular immunity, your B cells are part of your humoral immunity. So it uses antibodies that circulate through your body fluids to specifically target a pathogen. So with your B cells, they also need multiple stimulation from the pathogen. So at least two antigens will bind to the... Oh, shucks. That is wrong. Okay, let's try that again. So with your B cells, Still, homecoming committee, they can do stuff, but they still need student council's help or help from your T-cells. Still a part of your adaptive immunity squad, so still are specifically targeting a very specific pathogen, but they use it, the specificity is based on the antibodies that circulate through the body fluids. So with your B cells, in order to activate them, they bind to multiple antigens, which activates the B cells through multiple steps, and we'll cover those later, to make an army of clones to attack and remember. So unlike your T cells, your B cells are born and educated in the bone marrow. So they don't get sent to boarding school in the thymus. They get everything done in the bone marrow before they're deployed in your lymphatic tissues. So your B cells, after they're born, they're educated still in your bone marrow. They still undergo positive or negative selection if they fail. So positive selection, again, is where it has to display the surface antigen receptor. So with your B cells, a ton of those antigen receptors. It also cannot react to any self-antigens. And then if it fails, it also undergoes through clonal deletion or anergy to prevent those cells from reacting when deployed out of the bone marrow into your lymphatic tissues. So when your B cells have been deployed, the antigen receptors on the B cell surface, so it has multiple receptors on its surface, once those receptors bind to multiple antigens, so with your T cell, it only has to bind twice. With your B cells, they have to bind multiple times. Your B cells will then become antigen-presenting cells, so they'll take in those antigens, digest them, and display part of that cell, so part of the antigen, on its surface and become an antigen-presenting cell. So your B cells presents that antigen on its MHC2 proteins, so that's where it makes that little tag to be recognized by your T helper cells. So here, T helper cells will recognize that antigen on those MHC2 proteins and then it will bind to it. Once those T helper cells bind to that antigen MHC protein complex, so the antigen on the surface of that the B cell, it will secrete interleukins. Those interleukins are the signals to that B cell to make that B cell clone army. So that's where it needs the help from your T cells or the student council to actually do its jobs. So once the B cells receive the interleukin signal from the T cell, it undergoes mitosis to make the B cell clone army, so a bunch of copies of that B cell specifically targeted against that pathogen, so the antigens that it bound to make a bunch of either plasma cells or memory cells. So with your B cells, your plasma cells are the ones that create antibodies against that specific pathogen that the B cell bound to. So against that antigen of that pathogen. Your other cells become memory cells, which will circulate through the body and allow for a quicker response upon re-exposure to the same pathogen. So these memory cells, just like your memory cells for your T cells, allow for a quicker response the second time you get exposed to the exact same pathogen. So it's only for the pathogen that they have the shaped receptor for binding to that antigen. So your next type of cell, still with your upperclassmen, are your upperclassmen, still your lymphatic cells. You have your phagocytes, and then your structural cells. So your phagocytes, that's kind of like the jocks or your athletes. They eat a lot. That's their job. They phagocytize stuff, anything that's foreign, any kind of tissue debris, it makes contacts, it takes it in, and it eats it. Your dendritic cells are similar to your macrophages, except for they hang out in the skin library. So they're kind of like nerds. They stuck in the skin. They don't migrate around like other macrophages. They stay in one spot and study. So they still have a healthy appetite like your regular macrophages, but they only work in the skin. So you could say skin even at like a lab. So it's stuck in that one spot instead of being able to go around in different spots. And then lastly, for your lymphatic cells, you have your structure cells, so your reticular cells that make up the structure of your lymphatic tissues. So help keep them in their particular shape as lymph flows through them, which is fluid, so keeps this shape in this particular structure. Okay, moving on to our freshmen, our leukocytes or our white blood cells. So again, never let monkeys eat bananas. So your neutrophils, your lymphocytes, your monocytes, your eosinophils, as well as your basophils. So the job of your lymphocytes, they're kind of your overeager freshmen. So they can either kill by phagocytosis or respiratory burst. So for your neutrophils, the inflammatory chemicals that are released upon an infection causes them to stick to the endothelial cells of blood vessels, and then they'll squeeze through the gaps in between those endothelial cells. They're driven by chemotaxis, so increasing concentration of your inflammatory chemicals to the particular site of an infection. So with your neutrophils, I guess I didn't put this on here. So phagocytosis, we've covered this over and over again. The respiratory burst, that is where it releases your superoxide as well as causing your hydrogen peroxide to form. So these two super radicals, free radicals, will go and cause damage to other structures. So DNA, it can cause structures to other cells. So when it releases that burst or cloud of chemicals around it, it kills anything in the nearby vicinity. Your lymphocytes, so these are still white blood cells that will differentiate into your NK cells, your T cells, as well as your B cells. So they're kind of like waiting for their turn in the power spotlight. So the rest of your leukocytes, you have your monocytes, or as I call them, JV sports. So eventually they will mature into macrophages, or the other jocks, to phagocytize pathogens and tissue debris. So basically eating everything in sight. Then your last two, they have a little bit more specific function. So you have your eosinophils, or the weird kid. Their job is to fight off allergens and parasites. So their only type of lymphatic cell that actually deals with allergens and parasites. And then lastly, your basophils, they don't directly attack a pathogen, but they're very helpful. So kind of like the teacher's pet. Their job is to help the other leukocytes. So they'll secrete leukotrienes, histamine. So again, causing those endothelial cells to have bigger gaps in the capillary wall, so those white blood cells can escape out from circulation. And then also your heparin. Okay, now moving on to your antimicrobial proteins. So you have two types of antimicrobial proteins. You have your interferons and your complement proteins. So your interferons, I liken them to like newspaper. Their job is to alert that there is an infection. So the cells that are infected will secrete the interferons, which give the breaking news of an infection. So kind of ring the alarm. This is that there's an infection going on. So radicalized headlines just like a newspaper. For your complement proteins, I liken them to yearbook. So their job is to cover both adaptive and innate immunity. So they're kind of everywhere, not in a ring the alarm, something's happening kind of way. Their job is just to work within 3 different pathways to cover everything. So they cover both adaptive immunity, which is the ones that are specific to a pathogen, so you specifically can't sit with us, or your innate immunity, where the glycocalyx doesn't match the glycocalyx of the host body, so it's you all can't sit with us. So those 3 pathways with your complement proteins and how they work are your classical, alternative, and your lectin pathways. So for your classical pathway, it works through adaptive immunity. So these complement proteins will bind to a antigen antibody complex. So an antibody has bound to an antigen, and then a complement protein binds to that complex. When this happens, it brings more complement proteins to that antigen antibody complex in a process called complement fixation. For your alternative and lectin pathways, so working with innate immunity, where the cells recognize in general that this pathogen doesn't belong to the body, the complement proteins will bind to the antigen on the pathogen and then cause an increase in more complement proteins to be made. So more proteins that are your antimicrobial complement proteins. So the 4 ways your complement proteins will help to destroy a pathogen. So you have your 2 of them, your inflammation, so stimulating the secretion of histamine, so causing those blood vessels in your capillaries to enlarge, those gaps between your endothelial cells to become larger, as well as secreting other inflammatory chemicals, so to attract your neutrophils as well as your NK cells to the site. It's also involved with your immune clearance. So over here, where your complement protein forms a bridge between the antibody antigen complex on the pathogen, and then that bridge binds it to a red blood cell. So once that red blood cell circulates through the body, eventually reaching your spleen or liver, the complement protein as well as the antibody and the pathogen are all taken off the red blood cell. Your red blood cell is now free to go. It's not damaged in the process, but all that's removed off that red blood cell to allow your red blood cell to move on and your pathogen to be attacked in the liver and spleen